All right, so let's look at protein synthesis in this video. This would be the second video in the DNA unit, all right? And the big thing with protein synthesis and the fact that we need to talk about it at the same time as we talk about DNA is that is the point of DNA. DNA is like a cookbook that tells us how to make certain proteins. We're going to look at RNA compared to DNA in this video. We're going to talk about two different functions or two different processes. We're going to talk about transcription and we're going to talk about translation. Now, here are the central questions you want to be able to answer when we're done with this video, all right? You want to be able to tell me why we need RNA to make proteins. You want to be able to describe transcription and where it happens in the cell. You want to be able to describe translation and where it happens in the cell. You also want to know what a codon is and what the genetic code is translated to. How does that? How do codons and the genetic code? How are they related? Now here's the problem. DNA carries the code for making a protein. All right, we've established that. That's what genes are. Okay, the DNA is located in the nucleus. There are no ribosomes that are going to make proteins in the nucleus. So you got to somehow get the code from the nucleus out to the ribosomes, all right, because the DNA itself can't leave the nucleus. Well, here's the solution. We use RNA. When we talked about RNA in the last video, we said it's a single-stranded polypeptide, a single-stranded polymer or long chain, right? And this is the molecule that carries the code from the DNA in the nucleus out to the ribosomes in the cell. When you see the term RNA, it stands for ribonucleic acid, so RNA. Now, when we compare RNA and DNA, we actually see that there are quite a few similarities. They both have a sugar, okay, that is based on ribose. In DNA, it's deoxyribose. In RNA, it's just ribose. The structure is obviously different, okay? DNA is double-stranded, so we usually draw it. Let's see if I can get this pen up and running here. There we go. So we would draw it as a double helix, just like that. But for RNA, it's just a single strand, and so we draw it like that, like half of a ladder. The bases for DNA and RNA are almost identical. A, C, and G are present in both. The only difference is DNA has T and RNA has U. Now, RNA can be broken down into a couple different types. There are three types. There's mRNA, tRNA, and rRNA. mRNA carries the DNA code from the nucleus out to the ribosome. It's the messenger. Transfer RNA carries amino acids to the ribosomes. Remember that proteins, right? We learned this way back in the beginning of the year. The monomer of a protein is an amino acid. So you have to bring a bunch of individual amino acids to the ribosome in order to stick them all together to make a protein. So transfer RNA or tRNA carries the amino acids to the ribosome. And finally, there's ribosomal RNA, which is rRNA, which is part of the ribosome itself. We're really going to focus on the first two types of RNA in our class. Now, protein synthesis has two steps, okay? The first step is transcription, and the second step is translation. There's an easy way to remember which one comes first. Let's see if I can get a different color pen here. Let's go with, we'll say green today. Oh, no, that's terrible. Let's not go with green. Go with blue. All right. Transcription, translation. C comes before L. So after trans, which means across, transcript is script here, is to write, so to write across, and translate is to kind of move across, okay? C comes before L. So transcription is the first step, translation is the second step. The DNA code in transcription is going to be copied to mRNA. That way it can leave the nucleus. It's going to be copied, right? So to script or to scribe is to write. So we're copying the DNA code across the nucleus. We're getting it out of the nucleus. In translation, well, we're just going to translate mRNA into a protein. So we're going to read that mRNA copy and put the amino acids in the right order. So here's transcription. It looks a lot like DNA replication, except instead of making 2 copies on both sides, we're just making 1 copy. And you can see right here, this is the messenger RNA. And we know it's RNA because we don't see any T's. We only see U's, all right? And we know that uracil is the nitrogenous base. Sorry, something in my headphones there. It's the nitrogenous base that takes the place of T in an RNA strand. So the DNA is going to unzip, nucleotides are going to fall in and make an RNA strand, and then the mRNA is going to leave the nucleus through the nuclear pores that we learned about when we talked about organelles, and it's going to exit the nucleus and go to a ribosome somewhere out in the cytoplasm. It could be a free ribosome in the cytoplasm. It could be a bound ribosome on the rough endoplasmic reticulum. Either one is capable of translating an mRNA strand. Now, translation, okay, is pretty complicated. It's not something that it makes sense at first go because it has a couple different parts going on. So once the mRNA, so here's the mRNA. Start right here. Start with this picture. Here is part number, we'll say right here, part number 1. Here is our mRNA strand that we just brought from the nucleus. So this is from the nucleus. And it is going to be read by a ribosome. Now, when we say read, what we're talking about is a pretty complex chemical arrangement where the mRNA is going through the ribosome. Ribosomes have 2 parts, like subunits, and they kind of fit together like that. And the mRNA is fed through, and like a protein comes out the top, something like that, okay? And what the tRNA does is the tRNA is this thing right here. So all of this is the tRNA. And the tRNA has to be complementary to the mRNA. All right, what's that mean? Well, on the mRNA strand, so here's our strand, we have these 3 bases, the sequence. We have C, U, A. So we know that C always bonds to G, U always bonds to A, and A always bonds to U. So this is complementary. All right, and we can see that these sequence, they match. Now, attached to the tRNA, so again, this is my tRNA, this part, tRNA. Attached to it is an amino acid. Amino acid. And as each one of these falls into place, the next amino acid gets bonded, it gets tagged on to the chain. And so as you read the strand, so the strand's going this way into the ribosome. And so like these have all been read. These 3 were read, these 3 were read, these 3 were read, these 3 are going to be read, whatever. It just puts the next protein in the right order. The ribosome is kind of like a factory. It's got the right shape for those amino acids when they get close to each other to bond and form polypeptide bonds. So each one of these lines is a peptide bond, and this whole thing is a protein because each of these is an amino acid, right? Each one of those little boxes. So leucine and alanine and aspartame and or asparagine, sorry, not aspartame. Leucine, alanine, whatever. Okay? So let's break that down a little bit more. The mRNA code is going to be read 3 bases at a time. We call the 3-base sequence a codon. So let's go back. CUA right here, that's being read. That's a codon. The tRNA only attaches to the codon if it's complementary. We call the opposite side the anticodon. So that would be the anticodon. So the codon is a 3-letter sequence. The anticodon is a 3-letter sequence. They must be complementary. Every different kind of tRNA, 3-letter sequence tRNA, only carries one kind of amino acid. So everything's going to happen automatically because of this base pairing that's occurring. So these things are going to match up, and so boom, leucine is the 4th protein in the chain. Next, we're going to get GCG, so we're going to have C to G, G to C, and C to G. These are going to line up right there. And so now the next thing will be alanine, and that will be the 5th protein in the chain, and so on and so forth, building out your protein chain one amino acid at a time. Now, your tRNA sequence, I know that drawing of tRNA a second ago was pretty basic looking. The tRNA is actually shaped more kind of like this picture here. As it goes along and it drops these protein codes off, it'd be really useful if we could predict what's gonna, be our chain, what amino acids are going to be used. We can actually do that using this chart. So you read this chart, you have the first base in the codon right here on the left, the second base on the top, and then the last base is in the cylinder. So let's see if we can use this thing. So let's say we have UGU. All right. So U tells me I'm in this row, G tells me I'm in this column, and U tells me I'm right there. So UGU, right there. Boom. All right, so that's how I use that thing. Let's try it. So let's try another one. Let's say UCA. There's my sequence. So U, C, and then A tells me I'm the third one down. Serine. Yep, there it is. UCA, serine, right there. All right, we could do another one. We could say ACA. All right, A, so I'm right here. C, second column, A, threonine, right there. And you'll notice a lot of different sequences might code for the same thing. Like CU anything is leucine, right? CU and then just blank. So CU, well, it doesn't matter what I pick, I'm going to end up with leucine, okay? If I had CC something, well, I'm going to end up with proline, all right? What if we had, I don't know, G anything, all would be glycine, okay? So you can have more than one way to code for an amino acid. 3 really important ones are UAA and UAG, as well as UGA. All 3 of these are our terminators, our stop sequences. So UAA, UAG, or UGA. Those all mean stop. If you don't have stop, then your protein just keeps growing and growing and growing, and it becomes useless, okay? You got to stop it at the right length. So that's how you use one of these codon charts. So this would be a codon chart. And there's a lot of different ways that these charts can be utilized. They also make some that are spherical, which I actually like them better. They have like U, C, A, G. I think there's one of these in another one, and there's like a circle around this. And then it breaks the next row into U, C, A, oh, I missed one, and G, and then there's like one more row, and it says like A. So C, A would be histine. And then you just start in the middle and go out. So C, G, U. So C, G, U would be cysteine. There we go. Right. So it just kind of works its way out. So there's a lot of different charts that you might see all of them can be used to figure out what amino acid sequence you're going to end up with. So an overview of that. It's 3 steps, it's 3 parts. DNA is transcribed to mRNA. mRNA is translated to a protein. It's kind of like writing a sentence, right? DNA is the original language. We don't speak that language. We speak protein, not DNA. mRNA are our letters. Codons are the words. Amino acids are the words in the new language. And protein is the sentence in the new language. So I've got a, big DNA strand. We're going to copy it to mRNA. And we're going to turn it into a 3-dimensionally shaped protein. All right, that 3-dimensional shape is really important. Remember, when we learned about proteins, we talked about primary, secondary, tertiary, and quaternary structure. The shape of the protein really does matter. All right, so you should be able to now answer a lot of these questions. First, why do we need RNA to make proteins? Well, we've got to get it out of the nucleus, right? We can't get the instructions out of the nucleus without RNA. Transcription happens in the nucleus. It's the copying of DNA to mRNA. Translation happens on a ribosome, either in the rough ER or the cytoplasm, and it translates mRNA into a protein by using anticodons on tRNA to bring the right amino acids in the right order. Codons are 3-letter sequences, right? And the genetic code is the order of codons that gives us a specific protein. So a lot of stuff in that video, all right? Some potential questions you might see, all right? The purpose of protein synthesis. So why do we need to make proteins? What are the 2 steps of protein synthesis? That's something you might see. Might want to be able to describe what happens in transcription, what's a codon, and what the purpose of tRNA is, okay? So look over those. Those are some potential test questions you might see or review questions you might see, okay? And you should be able to answer them using information in this video. If you have questions, let me know. I'll be glad to